and graduated from chemistry at the University of Madras. And then subsequently, she went on to work for IIT Madras and Delhi. And as a chemist, probably she was one of those particular pioneering members where she tried to understand the informatics and reason data behind bioinformatics. That was how we can probably look into. And then, as a part of her work, as a part of her PG and postdoctoral, doctoral and postdoctoral works, she identified atomic screening constants, and then that was followed by works at France and as well as in New Orleans towards her PhD. And then she made a stint in Department of Chemistry at Jawaharlal Nehru University. She joined as an information scientist, and later in 1993, she moved back to University of Sydney and joined Australian National University as a professor. And she is the only bioinformatics, women bioinformatics chair at Macquarie University, Australia, or for that matter, across all the universities in Australia. And she has largely been associated with International Society for Computational Biology since 2003. And ever since she joined as an Australian director for the International Society for Computational Biology. She has prolific record of publications, I would say more than 250 to 300 publications. And she has recently won the prestigious International Society for Computational Biology Outstanding Contribution Award for the year 2023. She has several accolades apart from the ISCB. She has obtained Indian National Science Talent Scholarship for the years 1973 to 1982, and the French Government Scholarship from 83 to 84, and the UNESCO Chair of Biodiversity Informatics from 2004 to 2006. She also served as an elected fellow of the Abdus Salam International Center for Theoretical Physics from 1989 to 1991, and she was accorded the 2018 Australian Bioinformatics and Computational Biology Society Honorary Senior Fellow Award. And to the young tots like us, she obtained a first publication way back in 1982, which many of us here have not gone, or maybe we were just toddlers. She's known for several publication series. I'll speak two important publication series which we all should really take inspiration from. One is the Encyclopedia of Bioinformatics and Computational Biology series, for which she has been the editor-in-chief, and she's currently working towards a second edition of it. And when I was doing my PhD from 2004 to 2008, we went on to develop a hypothetical protein database, and that was when I came across one of her well-cherished papers called as MIABI guidelines, which speaks about minimum information about a bioinformatics investigation. So what happens to a database once say PhD fellow or postdoc leaves the lab? So how do we employ persistence or reproducibility or disambiguation? All those kind of things speaks laurels and volumes about that particular publication. And at least whenever I interact with students or whenever I mentor students, I speak specifically about this particular MIABI guidelines paper. And even for my ethics course that I teach, I put this particular paper as a part of our presentation. So with this little introduction, let's not take further time. So the program will be like this. Professor Shoba will speak for a little over 10 minutes, and then there will be some Q&A session extempore from me, and that will be followed by the audience here. Thank you so much, ma'am. So the floor is yours, please. You can start sharing the screen. Otherwise, I can share it for you. Yeah.
Okay, I actually had a bio in my slides, but Prash has been talking so much of my bio that I'm getting a little concerned about saying the same thing again. And I do apologize for the technical problems. Hopefully the Android phone works better than the PC. Or it's my PC is under CrowdStrike currently. I don't know.
Yeah, you
So,
could proceed. Just, what do you call, sharing the screen. Yeah, if you can see the screen, please.
okay.
Can you see the screen? Yeah.
Yeah. So I put together a few slides to show you the path that my career has taken and why I did what I did. Because I started out with quantum chemistry, which was later on called theoretical biochemistry. Then we were called biocomputing people. And then the new name came bioinformatics, but we are doing the same thing all the time. We have been working with computers. I started programming in 77, so it's a long time ago. So that's the kind of experience we have in this area. Prash, can you put on the next slide, please?
Yeah, please. Yeah.
Yeah. So after studies in India, including PhD, I went to France for postdoc and was a research associate in New Orleans. At that time, New Orleans had one of the largest computer centers, one of the 4 in USA. So since we need large computers, I went there. So then I came back to India and became a lecturer, but you know how it is, lots of teaching, hardly any time to do research. So I went back to teaching, to research, and was working in the Bioinformatics Center at JNU as information scientist, after which we moved to Australia. I migrated to Australia and worked as a research fellow at Sydney, University of Sydney, then in John Curtin School of Medical Research, and then also joined the Australian National Genomics Information Service, which is actually was like the NCBI in Australia. However, we didn't have the support of the federal government. So after some time, it sort of slowed down. Then I went to Singapore because they invited me to start the graduate program in bioinformatics. And then after a couple of years, I came back to Australia as the chair of bioinformatics. So what I'm trying to say is that we have to go where the opportunity is, because when you go to different labs, you understand how things work, how people are thinking about the problem. So it gives you great exposure. So maybe it's not a bad idea to move and acquire a little more knowledge. And Prash will tell you how his visit to Denmark was actually, like a light bulb moment and exposed him to so many other opportunities. So that's what I'd like to say. But nothing prevents you from working hard and working in a focused way. The important thing is to not do the same thing that everybody is doing. Maybe we move on to the next slide, Prash. My
Yeah.
first paper was in 1982. It was in quantum chemistry. So we moved on to maybe something like bioinformatics when I was in France. In the next slide, you can see that we worked on gramicydine channel. These were the days when we did not have video terminals, and we had to project everything on paper to see where the ion was going in the channel. So every time we used to print out and see whether the ion was still in the channel or had escaped. So it was very funny, but that's how we did it the hard way in the old days. Today's people don't have any trouble at all. Everything is on video terminal. You can even use 3D video terminals. You can use 3D vision. You can use immersive systems. We didn't have all those things. These are the old days when we just struggled with what we had and tried to make sense out of everything. So my exposure to New Orleans helped in many ways, because Pulitzer was the first person to think about electrostatic potentials on the molecular surface. And that has actually helped a lot in understanding how systems interact, how small molecules interact, as well as how large molecules associate. So protein-protein interactions are very much based on electrostatic interactions on the surface, and that potential is easily calculated. And that was a wonderful experience to understand the scientific basis of these things. Okay, then next slide, please. Okay. Then I've been going around in Asia, India, Australia, Singapore, Australia, and now again back. I'm a consultant of to Supercomputing Center in Singapore. But all along, what I would like to say is that we have to be proactive. We have to take the trouble to meet people and do things. We also have to do a lot of unpaid work. A lot of people say, why should we do this? Why should we do that? But we did all that very happily. And I've been with ISCB, International Society, since 2000. I've also been with Asia Pacific Bioinformatics Network since 2000. And I took it upon myself to spearhead bioinformatics in the Asia Pacific and assist raise awareness in many countries where they didn't have bioinformatics. So we've been there, we've started at the grassroots level, run training workshops, made sure that students who studied overseas and came back stayed there. Some of them are now leading research institutes, such as Sisajes Tongsima, whom I've known for since 2001. So we know lots of people because of that. We keep in touch and we help them, assist them where they can, and now they are assisting us. So that is how we are actually improving the state of knowledge in this region, because our people are very knowledgeable, they are very bright. However, they need to focus and they need to apply themselves in a way that actually makes them get world recognition. And that would what is really important, because then you're one among the world's elite scientists. So how to get there is the question. It means you have to, focus your work done. You also have to work very clearly and in a very focused manner, but also need to think outside the block. There's no need to do what everybody else is doing. You also have to study the problem, read a lot, and come out with a totally novel solution. Because biology is not complicated, it's fairly simple, it's straightforward. Maybe we are making it more complicated than it because we are forcing it to follow our rules. So think of a simple solution. A molecule has to go and find an enzyme. It's just swimming around. It's a tiny little molecule going through the bloodstream. So the recognition mechanism has to be simple, but very specific. So how can we do that? We can do that because we have to think like biologists. Then we can do really, really well. Okay, whereas what's in the next slide?
Yeah, please.
So, okay, I worked in many different research areas, and many people have always asked me why I didn't specialize. I did specialize. I specialized in all of them. The thing is, things are interesting, so I do them. And we are like a computer, so we can do multiple areas at the same time. But the challenge is to keep up to date with all the literature and the latest findings so we can actually lead the way. So I never had a team of students where all of them did the same project. Everybody did a different project. And I was with them and they fit into each other. They criticized each other, they worked together, they supported each other, and they all learned different things. So that's how my team has been working. We are not like biologists where everybody is mutating a different amino acid on the same enzyme. We are doing totally random things. And many of the problems were brought to us by people who generate the data, the biologists themselves, because they ask, how do we solve this problem? So then we start working on it. I believe that unless our science gets practical application, it might die. So we need to make sure that what we produce as research has a home and leads to some solutions. So otherwise, we will be in our own ivory towers and we will be locked out. So we need to be able to assist, apply, and use our knowledge to the benefit of science, community, and humanity. So that would be the approach that you need to think about. So the next slide, I think, is the last slide, I hope. So for all these years of struggling along, I have managed to get some awards. The Australian Society gave me a fellowship award in 2018. And I've been working with Asia Pacific Bioinformatics in a network for a very, very long time. For as president, vice president 2 years, president for 12 years. And then I said, I'm not going to do this anymore. We need some young people. Maybe Biden should have listened to me a long time ago. So I stepped down and made sure that the society continues. And I'm glad to say that one of the people for whom I was the thesis examiner is currently the president. And we are doing well and everything is going well. So finally, the International Society realized that, they are lacking some awards in our direction. So they elected me a fellow last year and gave me the Outstanding Contributions Award as well. So that's not the end of it. We are still continuing to do things for them and with them. So we hope that you can join us this year in our flagship conference, INCOB, which is going to be held jointly for the first time in Japan. We spearheaded this in 2018, but then were hit by COVID. And so after that, everything became slow. So finally, the conference is going to be held in 2024. The idea was to unite all the scientists working in bioinformatics in one location, rather than have 5 different conferences and not everybody can go to every conference. So the idea was to have a super conference which integrated GIW and INCOB, which are the main 2 conferences in our region, plus all the little ones who are not actually big contributors, but they have said, okay, they will not hold any conference this year and they will join us and partner us. And this is APBC and also the Australian Society. So they're all partners. So they're all going to hopefully turn up at this meeting. It is also ISCB Asia, and therefore it will be a great opportunity for all of us to meet. So I trust you will go there and meet all your colleagues. And that's it. I'm done. So I'm open for questions.
Thank you. Thank you so much, Professor Shoba, for that scintillating talk. Especially one of those particular sentences that you say that, we should honor and welcome the unpaid jobs. That is a very important thing. And I think, the youngsters or the current generation, could really, honor that kind of commitment, which is certainly the need of the hour. So moving further, Professor Shoba, the very first question that I wanted to ask is, as a chemist, what were the initial hiccups that you faced, facing bioinformatics? I mean, how did it all set it, from chemistry to bioinformatics? Yeah.
Chemistry is a hard subject. It's a bit like maths and physics. So it's either plus or minus. It's not anywhere in between. Whereas in biology, you have every shade of gray. So, and there are no real rules. Chemistry is full of rules. Things only happen in a particular way. But in biology, it's all a bit fuzzy logic. So it's a different learning thing, but we have to understand what the problem is. And once you do that, it's a lot easier. I feel that my training as a chemist actually empowered me to understand how mechanism works, how function works, how recognition works, and also in the context of what would happen when a mutation occurs, because it is actually the chemical nature of the substitution that makes the difference. So I think that helped me a lot. But we always have to think that biology permits a lot of changes. Not every change is lethal. Not every change is important, because many changes can be compensated as well, even if they are critical. If one enzyme does not work, we always have a backup plan. We have many enzymes that can step in and maybe do a 50% job. So the organism doesn't die. Maybe it's a bit slow, but things happen. So rarely do things actually go critically wrong that makes a difference. So when something like cancer hits, it means several thresholds have been crossed. Several steps have occurred And only then we are able to figure out that somebody's got a disease. So I have been thinking about how to solve this problem by, say, using liquid biopsies, and how to do this well ahead of the person actually manifesting the physical nature of the disease. So there must be a slow progression which we are not not capturing with the sensitivity of our current methods. So, how could we get there? That would be a very interesting question. Is it to do with the sensitivity of the instrumentation, or are we not doing the correct tests? Or are we not doing them frequently enough to notice a change? And not all changes are going to be statistically significant. That is something we must keep at the back of our mind. Whatever is statistically significant is usually downstream effects. Those are housekeeping genes, and they don't matter. So we need to figure out how we can overcome all these biases that are already inbuilt in our system, and to focus on those few things, simple changes that could actually be amplified further down. So, how can we get there? And that will be the challenge to all of you who are the young scientists of today.
Thank you, Professor Shoba. Absolutely, very well said. In fact, in the very first slide, the question and answer, from the audience will be later, please. This is in Sciversation, so there will be.
So,
please.
Jay Kishan is asking a question, I think.
Professor Shoba, we'll take those questions later, please. I have few questions now with the permission. Please. So the very first slide, when you were talking about the electrostatic interactions, that was that caught my eye, that caught my attention, because today I was just brushing up my things, and my 12th grader, I was just trying to tell it to my daughter how electrostatic interactions could really sum up ascertaining the protein interactions and all those kind of things. But during those days, almost 40 years ago, when you started working towards this, bioinformatics has evolved so much. I mean, and yet you were not only jack of all trades, but also mastered across all these multifaceted disciplines. What were the initial challenges that you could surmount, Professor Shoba? Could you just give one or two small examples to the young audience here? Taking one example, maybe, if you have posed any problems, how could you convert those problems into opportunities or successes?
So some of the studies we have done have been only because of experimentalists. One of them was the number of EST studies we did with the helminth parasites. It's because a colleague of mine, Robin Gasser, who's in Melbourne, he asked me, how do we do this? We're generating so many sequences, but we have absolutely no idea how to analyze them. So can you help us? Then we started putting things together, making workflows. We found that there was no need to invent the wheel, because every step of the analysis had actually been thought out. So we actually published something called The Hitchhiker's Guide to EST Analysis a long time ago. And Shiv Shankar, who authored that, who's the student who worked on that, revised it, I think, 23 times. He was totally mad at me. And he kept telling me, when is it going to be done? So I told him it will be done when it's ready. So he did not understand that, but now he's an associate professor in QUT, and I'm sure he understands that and is following that. Because it's the completion of something and bringing it to a level where it's really of high quality that makes a difference. And when we published that in Briefings in Bioinformatics, that journal actually had an impact factor of 24.3. It was on a sticker on the cover, and I remember him taking a photograph of it. So you see, we have worked very hard to get where we are, but you should not think that you are not capable of it. But don't be in a rush to submit things that are half done, because it creates a really bad impression. And then they seem to feel, oh, scientists from this country are no good. And then that tag stays on. So it's important to convey your message very well and take trouble over it. So this is what we did. So
Wonderful, Professor. In fact, the Hitchhiker's Guide was in my list. That was my next question actually. But you stole that. That was a wonderful. In fact, I was just
So-
submitting my thesis in 2008, and in 2007, I believe this paper came up. And my mentor and my supervisor was telling me, "Hey, Prash, did you look into this?" Because I was then, annotating a large number of hypothetical proteins, and we were largely working into the express sequence tags. And that paper also caught my attention. That was a wonderful paper. Yeah, yeah. Thank you so much. Yeah.
So
Yeah.
we also realized when we published that it's important to convey your message in a very catchy way.
Yeah.
And what happened was that we are trained in India to write these terribly long sentences, about one para long. And the Western world does not understand such complicated English.
Right.
So, although we are a non English speaking country, basically, we are all speaking our own languages. It seems we have been so well taught in English that we are writing in some ancient style. So, it's very important to make sure your sentences are only 10 words long, because after that, your reviewers don't understand what's happening. So, please make it brief, make it straightforward, and try to use active voice, not passive voice all the time,
Right.
because then you're conveying the message quickly and efficiently. The second thing is you need a very well crafted abstract, because your abstract is what is published in all the abstracting journals, including PubMed. So if your abstract is not interesting, nobody is going to read that paper.
Right.
So please make it abstract and have a catchy title. We tried so many titles, and finally I said we'll write Hitchhiker's Guide. So that looked good, so we did that. So we need to have a way to express ourselves. We just need to try. We can try. We are good at writing, so let's try new words. That's all. And we recently published a paper in which we used the invisibility cloak for cancer,
Right.
how cancer hides from the immune cells. So we used Harry Potter's term. So we can be, we can make it conversational and interesting. That's all I'm trying to say. So if you make it interesting, people actually look at your paper, they read it. Okay, so that's the catch to this. I'm happy to help people who need some clues or assistance. It's like a quiz.
Yeah, thank you, Professor Shoba. And moving further, I was also amazed about your signal peptide database paper that also came up way back in 2005. That also we were using along with Raghav's tools. And of course, I was also in Denmark. We were also using Surendranak's tool and other things. What was the idea behind that signal peptide database tool, Professor Shoba? In fact, at that time, I remember there were hardly one or two tools that could predict signal peptides. So what was going through your mind at that point of time?
So the signal peptide is actually important in that it is a very conserved part of the sequence, and the rest of the sequence is sometimes different. The idea is the signal peptide actually provides localization. Where is your sequence going? So that was what was interesting to us. And actually, I'm glad you asked that question, because I remember how many times we struggled to get that paper published, because we had Rolf Epweiler in Uniprot sitting in EBI, who kept saying that we were wrong and Uniprot was so correct. Whereas most of their annotations were wrong. They were by analogy or homology rather than by experiment. So when we said only 10% of your data is experimentally validated, they were very upset. So we provided them every single one of those papers that had the experimental details, and only then it took like 8 rounds. So finally it got published. So you see, we have to struggle. We figured that out. But we are actually spearheading the way, because now everybody is talking about fake data and databases.
Right.
These are actually data that are there because of similarity to some other sequence. Many of the data have not been experimentally validated, especially the signal peptide, because the cleavage has to be observed biochemically and then the N terminus has to be sequenced.
Right.
So
Wonderful.
that led on to our caspase problem because it's similar to that. Like we have the caspase issue, and when one cleavage site has been cut off, then the protein unfolds and the next cleavage site is available. So we did sequential cleavaging and we showed that it's possible to observe these. Again, people are a bit uh,
Yeah.
not so happy with our results, but finally, experiments did show that it was possible if you wait long enough, you can observe subsequent cleavages.
Right. One final couple of questions, Professor Shoba. I mean, as a chemist, I thought of asking you. I have, in fact, a very fair intuition that we in India have produced large number of chemists in the world than anyone else, and yet, we could not probably reach somewhere near tertiary structures or quaternary structures, and probably, deliver those particular structures that are done through computer-aided drug designing and onto the market as proficiently as probably as we were to expect or we were probably capable of. So do you have any quick say on this, please? I mean, because now if you really talk about bioinformatics, 80% of bioinformatics is largely from India, chemoinformatics, basically. And yet, we could not produce those many good number of structures for the market. So do you have any quick say on this, please?
I'm not sure what the situation is now regarding funding for these projects. And also, many of these things have to be experimentally validated. So once you make a computational prediction, it's important to have a good collaborator who can do the experimental work. I'm not well trained in fact, when I went to university, we didn't even have molecular biology as a course. So I can't say that I'm trained in that. Maybe I could learn, but I haven't come down to that. So we need actually to validate these things. We have a large number of drug companies in India, and I'm sure they are doing computer-aided drug design, but it's being done in-house, so you don't hear about it. I'm sure a lot of it is coming to the market, but we don't know.
Right.
But drug discovery is a very expensive proposition, and it takes a very long time to get a drug into the market. So you need to put in $1 billion or something, which is almost impossible to do. Plus, you need to do clinical trials, you need to do mouse trials, get them validated, then you do the human clinical trials, and then it goes on a couple of phases, and then only it's approved. And the biggest challenge is that everybody is waiting for FDA to approve before we are even thinking about the drug. So that's another problem that we face. But I know many scientists in India who are actually working on natural products which have an Ayurvedic origin, and they are using that, isolating the compounds and testing and finding their efficacy against various diseases. So that is something. We have had people in INCOB who were initially there but are too busy. Durai Sundar, for instance, he's been working a lot on Indian medicinal herbs and their derivatives, the chemical compounds derived from them, looking for their biological activity and how they can be repurposed. Of course, if you're using a natural product, you cannot patent it. That's another issue. However, if you could use cheminformatics and look for similar molecules or even design one and then test it out, then you have a molecule that you can patent. So these are the things that you can think about. Cheminformatics is very possible. We have done a couple of theses already, and we have picked molecules based on AI. and shown that they are of higher efficacy than the original natural ligands. So it's possible, and we can do that. Now we are working on insect odorant receptors and looking for molecules that we can target for attracting insects, and so that this is called the biological pest control, rather than spraying everything with chemicals that cause a lot of environmental damage. So that's something that we are looking into. It also prevents crop loss and helps agricultural produce to be organic, and that's a good thing to do. So that might be something of interest to you people.
Right. Yeah, thank you so much, Professor Shoba. I'll just probably ask one final question before I let the audience speak. I was just wondering, how did you put your disagreements in an agreeable way, with your colleagues or maybe with your students or with your mentors at any given point of time? Because this is a quite common most question that I usually ask in Sciversation, because the current generation probably, with due respect to them, there is a lot of impatience prevailing with them. They may not probably disagree in an agreeable way on any aspects of science that they discuss, or any kinds of mentoring activities that they are a part of that. So, what advice probably you would give to the young audience if that were the case, to put any disagreements at any point of time?
So not every student needs mentoring. I have had students who took the problem, disappeared, and then came back with a thesis. So that's also possible. Some students need mentoring, some students need guidance, but definitely when the writing stage comes, they do need some interaction. We are not always the best way to say it. I let my students take the lead to write the draft and try not to change their language completely. But I do try to advise them because of the problems of examiners and reviewers not being able to understand them. It's not I can understand them, I can take the time, but not everybody has that patience or that approach. So we are only making them more globally competitive by helping them to present their work in a proper way. That's all. That's when I do more mentoring. And usually I have them sitting next to me. And whatever change I make, I tell them I'm going to use track changes. And you have the right to either accept or not accept. It's up to you. And you can also rephrase everything, because if you think that I've made too many corrections, simply remove the sentence and rewrite it. So I'll let them take the lead, because once they are a PhD candidate or a graduate, they are a scientist. So how are we going to get them there? So our job is to get them to that stage and provide them the steps. And it's their choice which way they want to go. So that's what I do. And it's up to them. So it's the students who have to be taking the lead in this. Some people write very well, some people don't like to write, some people write very cryptically. So I try to help them and give them some guidance, give them some example papers to look at so they understand how to write. That's all. It's not up to me, but I just facilitate that.
Wonderful, Professor Shoba. Friends, that was Professor Shoba Ranganathan, and I've learned a lot of things from her. She has, been a great mentor, a great scientist, and above all, a wonderful human being. And I had an opportunity to meet her at least 3 more times during my lifetime. Learned a lot of things, even the latest encyclopedia articles that she set on, for this. Editing stage, over 1,000 articles, which is not a kind of an easy thing, to edit them. She has been, the driving force behind that. And most importantly, she has obliged in giving her time today. So maybe we can request her to spend another 10 more minutes if anyone has any questions for the Q&A session. So please feel free to raise your hand and or ask your question in the chat box. But I would appreciate if you can quickly switch on your camera, introduce yourself quickly, and ask any question. Jai Kishan, yeah, you had some question, please. Yeah, yeah,
Hello.
yeah.
Yes.
Please switch on the camera, Jai Kishan, please. Yeah,
Sir, I'm connected to desktop and I don't have access to
yeah, please.
camera.
Yeah, it's okay.
So, so, hello,
Hello.
Shoba ma'am. Uh,
Yes, please.
it was a great journey. Like your throughout the life is inspiring young mind like me. I would like to ask you. So far, you have experience equally to my age, like the age I have lived. I I think we the big challenge in our area right now is storage as well as the computing power to process the data. So I I was hearing a lot of thing about we can use a DNA in future as a storage unit, and we can use quantum computing to process this data. So you have any remarks on this area, like what could be the future of storage device as well as the uh, process like a computing power facilities in a future?
Uh, there will always be bigger computers coming up, and we are also generating more data, so both of them are competing with each other. We have very good facilities in India at NIC, I think, and also a lot of regional computational facilities. But one thing we realized, we used to take condemned computers from the computer science department from their labs, stick in a GPU card, and run most of our processing because papers can't wait for queues. And sometimes you put it in a national system, you're waiting forever for things to finish. And that doesn't help always. So you could actually try these things locally. Get some old computers, buy compatible GPU cards that costs a fraction compared to buying the computer. Have two or three and try to process yourself. All possible. Using DNA as a storage device has been in the pipeline for about 40 years. We are still to see any devices that actually use DNA as for storage. It's going to be a long time away. So I think we will be waiting on that one. But we now have very good disks. The solid state disks are very good, so you could try and get one. And that would be a wonderful way to have a backup plan. The important thing is that the data, the results you get are very small. It's the intermediate processing that accumulates a lot of data. So if you can actually have the input parameters and store only the final result, you could calculate the rest on your own. That's possible anytime. So you don't have to store everything. And remember, once you've done the storage, you need to clear it, clean up. Then you will have more room. So that's something that has to be done practically. So and also try to focus and narrow down your search space. After a couple of rounds, you don't have to run your query on the entire data set. You need to start thinking about specific problems or specific areas of interest. Then you don't have that much data. So my theory is that we need more brain power than we need computer power. Because if you're smart, you will find a solution to all these problems. So let's think about it strategically and you will be able to solve it. And
Thank you.
also lean on friends and go and put your jobs in different places.
Wonderful, Professor Shoba. Yes, so you have a question, please unmute your microphone and quickly introduce yourselves and ask.
Hello, Professor. Hello, ma'am. I hope
Yes,
last year the same date we was together in NUS,
yes,
Singapore. Yeah,
yes,
I hope you remember. So, Professor, I have one question, like quick question. Like, as
yes.
you told, like the research we can't publish as soon as possible because we need to work hard and like it's a bad impression that if we submit earlier. But like in the case of like Professor, if you think like when we are going for the any like COVID-19 and we have found something on the COVID-19, like in the case of chemoinformatics we are just discussing right now. So like instead of going to only the docking and simulation, if we go hard like with the free energy calculation and all the thing, but on the same time maybe some person going to publish with like fast with me because this is like emergency diseases. So in
No, no,
these
no.
cases,
I did not say you should not publish quickly. All I said was your submission, your first submission of your paper should be well written. That's where we differ, not in the public. We do need speed of publication, but the quality of the submission is also very important. Otherwise, it doesn't lead to getting it reviewed because the editor has a serious role to play in this. So editors look at it quickly and then they say, okay, this is okay to go for review. Otherwise, they reject it straight away. You should publish quickly, but when you write, it has to be written well. That's what I said. When we wrote a review, there's really not any speed or anything there. It's only a literature review and a benchmarking study that we presented in that Hitchhiker's Guide. But what we did do was we wrote it in a way that it actually encapsulated all the issues that people were facing in one shot. So it took some time to write it, not that it wasn't on time, but it needed a lot of revision to get it to that standard. So when you read it, actually you understand everything. That's what's important. So sometimes it doesn't come across that way when you're a scientist because you know everything. So you don't need to write all the in-between sentences or the explanations. You just write something quickly and that doesn't help. Okay, somebody asked a question online.
Ma'am, I have one small question, ma'am. Like, as
Disappear.
you told, like one of your student did one project like 23 time revision. So, like, if I
No,
start one,
not a project, just that first review, only
okay.
one paper.
Okay, ma'am. Like for me, though, like if I have some good idea, but the thing is, if I think that it will take some more time, like not even like we can't finish in 3 years. So, like, do you think should we go for the like any PhD work with those topic, or should I wait for like after finishing PhD, I will start with my own? What do you think? Like if I have some good project and I think that we can't finish in 3 years, should I go with that project for my PhD or not?
If you can't finish in 3 years, then that's not a good project to take for PhD because after 3 years, your thesis is due. So you need to think about that. And your supervisor will actually be able to guide you best on the choice of the project to be selected in such a way so that you will be able to complete on time. Okay? And
Okay.
such blue sky projects are usually not given for PhD students. Because a project that requires 6 years or 10 years of work is best done as a hobby during a PhD, and afterwards, after you complete, you can take it up as your postdoctoral work.
All right, ma'am. I can understand. Thank you, ma'am.
So,
Thank you, Son. Thank you, Jai Kishan. Jai Kishan, I'll reserve that question later. If maybe if someone has any other questions. Any other questions, please, from the audience?
I saw a question flash and then it disappeared, so I
Well,
can't see it anymore.
It's
I
still
don't know
there.
where the chat box is in this.
No, it's still there, Professor Shoba. I can read it. I just thought if there are some more
Okay,
questions from
okay.
others. Yeah.
Okay, all right, okay.
So if there are no questions from others, I'll take that Jai Kishan's question. So he asks, can you have quick remark on the evolution of force field like? If I compare with programming language, we come along from Perl to Python. So is it possible to come up with force field writing in English-like language?
Actually, I don't write in Perl or Python, so I cannot actually answer that question. I was trained in Fortran. And although I learned Pascal and C, I'm unable to program in any of those languages because I go to default.
Yeah.
So I am not a person that you can talk to about programming because I am not in one of these scripting languages. Fortran actually is the best language for scientific programming. So if you learn that and you actually manage to write the code and it compiled, it executes the fastest. So if you're doing benchmarking, you should use Fortran or C. If you're doing just study, then you can use all these Perl, Python, R, etc. That's fine. But if you are benchmarking a program for speed, computational power, for precision, then you do need to learn one of these languages. So that's something that's a bit tough. And I do not know, you need a little more programming skills for that. Because anybody can write Perl and Python, because they can't write the other languages because they are stricter and they are more structured. But then they are faster. So that's the issue here. So many of the old code like quantum chemistry, molecular dynamics is still in Fortran. In fact, Modular was written by Sally in Fortran. But he went and had a tie-up with a company which made it academically inaccessible. So he rewrote the code in C. I sort of clued him on that, and he rewrote the program in C. And so now it's available to everybody. And you can also use Python to address it for scripting. That's the front end. It's not actually the back end. The back end is still written in the hard old languages, which can compute quickly. So there are a number of things that we are using here because we are using machine language and we are calling machine subroutines, which are already within the system. So if you learned one of those languages, you actually generate faster code. It only matters if you're doing benchmarking of large. If you're just doing, processing or making a workflow, then any of these languages are good enough, is fine. Because they won't cost you much.
Hello. So, ma'am, it's not about the programming which I'm asking. It's about force field which we are using. So each force field have its limitation, right? So is it
Force fields, unfortunately, were made in the 80s or 70s, and they have not been updated.
Yeah, exactly.
Because,
That's
because
what
there is no funding agency that's funding that. So if you want to do it for your research, that would be great. But you need to find the funding for it. So that's the only problem with the force fields. And force fields also are still being using van der Waals surfaces. And van der Waals surfaces, as we know, are based on atoms. And they don't actually have the depolarization and all those effects put in. So it's much more complicated than that, and actually you should use electric field. That is another thing that Pulitzer worked on, and that was his claim to fame. Using an electric field surface, 0.2 electric field encapsulates 99% of the electron density. So if you want to use that, but then you have to learn quantum chemistry and how to generate that. And that surface is difficult to generate. So some programs do it. There is a program called ICM by Molsoft. They use that. That's why their docking is one of the best in the world. So if you looked at many of their old papers, they actually cite this paper, Pulitzer's paper, where they use electric field. So you could use electric field and redo the force fields, but it's going to be a long way away. And then you need to show that you are as good as the others. It can be parallelized. It can be benchmarked against other programs. And we have many programs already running around, GROMACS, Amber, CharmM, and a few more. So whichever one you want to compete with, please go ahead and maybe you'll get the Nobel Prize.
Thank you so much, Professor Shoba. I hope that answers your question, Jay Kishan. Friends, without any hesitation, I think, we had a wonderful, evening today. I think, you all will probably agree. And I request all of you to switch on your camera, unmute your microphone, and give a big thumping round of applause to Professor Shoba. And also give a standing ovation to her, for her, amazing, contribution to the science, and as well as mathematics.
Thank you very much, everyone. But we still have to work hard every day, and that's what I'm doing. Okay, so
friends,
it's part of the game. So You are all capable of even better achievements because now you have the technology and you have the knowledge and you have the internet. We didn't have many of those things when we started out. So now make good use of what you have and achieve, much more than I have ever even thought about. Okay. That would be my challenge to you.
Thank you so much, Professor Shoba. May I request all of
Thank
you
you.
to switch on your camera, please? We'll have a good photograph with Professor Shoba. Unfortunately, some of you have not switched on your camera. I don't have an AI to switch on your camera, please. Yeah. And Professor Shoba, with your presence, we had over 54 participants today, making it.
Okay.
In fact, I've been organizing Sciversations over the last 4.5 years, but we never had this much audience so far.
Yeah.
That has also been a kind of a worrisome factor for us because we have over 9,500 members in Bioclues. We send an invitation and hardly 15 or 20 people attend. But today, you stole the show. Thank you so much. Yeah. And
Must be they're having a good time off today. That's why.
yeah. And I always say there's a good photographer in Kerala, and that is me, Prash. So I'm taking one photograph. Yeah. Yes, it is coming up well, but there are no smiles. I don't see any smiley faces. Yeah. Visu, can you please switch on your camera? And others, Sandeep Seni, Vikas, Swaleha, Prabhanjan. This is going to be a very amazing thing. Yeah. Thank you so much, Professor Samshurao sir and Professor Nirmal Loya sir for joining. Ma'am, if I may just introduce Professor Samshurao is the Vice Chancellor of Mangalyatan University in Jabalpur. He was also Vice Chancellor at Mizoram University. And we have Professor Nirmal Loya, sir needs no mention, he's also my mentor. He's currently based in the States. He's an emeritus at University of Rajasthan, at Department of Zoology. And he's also a Bioclues Lifetime Achievement Awardee for this year. Yeah. So thank you so much. So I've taken a good number of photographs, but I'll take one more, please. Yeah. Yes. Thank you. Thank you so much. And let's again give a big thumping round of applause. And all of you, please stay back. I just wanted to talk to you for 2 minutes, please. Huh? Thank you. Thank you, Professor Shoba. So friends, thank you so much for joining, all of you. I think it's been absolutely incredible. I just wanted to tell you a few quick things. We have been organizing such events over the last 20 years. We are celebrating 20th year this year for Bioclues. And you all received these messages, and unfortunately, not all of you attend these particular things. This time I had to push you. Please attend करके मैं कम से कम 3 बार मैं personally मैं message किया था. I had contacts of almost 350 people in my mobile phone. I had to send individually, please attend करके. Okay. But you can be very good listeners. We are not running any drama. We are not running any singing or a dance show here. We are speaking science. We are letting people speak science so that we can take inspiration from them. Okay. We have two exciting civilizations also coming up this year. We have Nobel laureates, 2 Nobel laureates who are also a part of this civilization. Okay. And you all have been a part of Bioclues. No matter what, you might be very busy. You can attend these particular things, interact with people. Professor Shoba has been a doyen, in bioinformatics. After all, this is for you. This is for free. Okay. And if you think that, you want certificates for such kind of things, probably, that is not acceptable. I know many of you vie for certificates. इसके लिए कुछ चीज के लिए कुछ certificates पाया नहीं जाता. Okay. We don't work for certificates. As Professor Shoba says, there were days where she worked for unpaid jobs. This is also my unpaid job right now. Okay. This is also the job that we do after 5 o'clock. This is also an unpaid job, but we learn a lot. We come up with some kind of learning experiences. Okay. For example, when I started writing my first paper in 2007, as a part of my PhD, I was the only first author in my particular paper. My supervisor said to that, Prash, I don't understand what you write. And then I don't know whom to contact. And then Professor Shoba said to that, okay, I'm going to review this. So she was the first one. She said to that at that point of time, peer review was just coming up online. And then, I could publish my paper, hip human proteins, in bioinformation, which has an impact factor of 1.8. That was my first paper. Because people like her who volunteered, who helped, so they were the people, who could really help us, move forward. We are still learning, from these kind of, things. And whenever you attend such kind of, events, kindly, switch on your camera, have a big smile. Okay, who knows, with your big smile, they might stay a few more minutes. They might be excited to interact with you more often. Okay, these kind of, things are etiquette building, that I could suggest you. Okay, this is where, the demeanor and etiquette, comes into picture. And I think, we're all really forgetting these kind of things. I had a great opportunity to interact with people like her. When I was doing my PhD, I used to very often, go to conferences, meet there, ask questions, like what probably Jay Kishan and Som had asked us today. Okay, keep away your shy genes and all those, kind of things. Okay, because seldom we get such kind of opportunities. Seldom, we get such kind of opportunities to interact as well. And the APJBC conference, Asia Pacific Joint Bioinformatics Conference that she announced, we at Bioclues are also a part of that particular partner. And we are thinking of giving away some kind of student incentives to life members of Bioclues to go and attend that kind of conference. And for Bioclues members, there is a huge discount as well for that. Okay, so I'll probably stop here. I'll not bore you much, but I just wanted to request all of you with two folded hands that Bioclues has been incepted in 2005 just for the heck of a reason, to ensure that we all grow together. Okay, and if you are not growing together, then there's something wrong. And we are not asking anything. We are only asking members like you to join forces, join hands together, learn whatever you have learned, and pass on the baton to the next generation. Okay, and if you are not able to do that, then probably we are not humans. We are inhumans. Okay, I'm not trying to pinpoint anyone. Okay, we are in the era of artificial intelligence. Maybe with a single click we might help from ChatGPT, but we may not get that kind of mentoring support which we are being trying to strive for. Okay, so with these little words, I'll probably stop all of you here. Thank you so much for joining. And I was thinking that we should have at least 50 today, and we got 54. Yeah, Professor Nirmal Loya, sir, please. Yeah. So please unmute your microphone, sir. You're muted. Yeah.
Can I take 1 minute? After listening to Prash, I could not resist saying few things. It has been a wonderful experience listening to Professor Shoba and her scientific journey and accomplishments, and organizing such events so regularly. It is a matter of great compliments, and I greatly admire Professor Prasant for taking pains of organizing such events, identifying very distinguished peoples in the field who are pursuing research in different aspects, and I'm sure that it is greatly benefiting those who are participating. And I also accept that large number of people should come forward and join such events. Thank you so much, Prasant, for giving opportunity to be
Thank you
part
so
of
much, sir. Thank you so much, sir. Thank you so much for joining. Pretty early in the morning. Professor Loya sir is in the States. It's 7 o'clock in the morning for him when the program started, and he joined. Yeah. So thank you so much. Yeah. I'll probably we are all put up at bioclues.org. You got the list of members as well. Chitra, Pratha, Mai. You can go to bioclues.org. The page is for you. You go to membership page. You have the list of all life members of Bioclues. And if you go to bioclues at googlegroups.com and LinkedIn, we have over 9,000 members over there. Okay. So you can probably interact with them. Otherwise, you can probably mail me. Yeah. So Santosh, yeah, you have any question, please?
Prash, in my university, the Bioclues website has been blocked by our firewall.
Oh, I see.
So how I can navigate like?
I think you should. I'm sorry for that. You should tell your IT admin saying that Bioclues is not a spam organization.
Okay.
It's a good thing, and you can tell that. Okay.
Okay.
That's very unfortunate. Yeah.
Thank you.
Maybe they your which university is it, Santosh?
I am from Kuvempu University from Karnataka.
Oh, I think you have to tell your Kuvempu University. You can ask your IT admin, mail him and CC me.
Okay.
Then we will prove ourselves that we are not spammers.
Okay, fine.
We are only people who work for society.
Okay.
And that way we can do that. Many universities and does Times of India work for you? Times of India website work for you?
Times of India. It works for me.
Yeah, that's very unfortunate, isn't it? It's a spam website, and we at Bioclues are not spammers. Sorry for the sarcasm.
Yeah. I can understand.
I don't mind lambasting your IT admin for this. If they have blocked such websites, I'm really sorry for this.
Yeah,
I'm very open and pluralistic on this. Yeah.
And I have recommended and I have added almost 30 members this year for Bioclues.
Thank you. Thank you so much. Thank you so much, Santosh, for this.
Yeah.
Thank you. Yeah. So if there are no questions, I think we should call it today. Thank you so much, everyone. Thank you, everyone.
Thank you, sir.
Thank you all. Bye.
Thank you.
Thank you all.